Thank you. I hope everyone can hear me. If not, just wave your arms and yell. I'm going to try and get through this a little bit quicker so you don't lose too much time in the day. But if you have any questions at the end, make sure to ask them. So as Odetta said, I'm here to talk about a brief introduction to characterizing organic matter with high-res mass spectrometry. I'm a staff scientist here at EMSL, been here for about 5 years, and a lot of interest in method development for this sort of characterization. And then this afternoon, there's a tutorial in which I'll expand a lot more on some of the information here. So what I want to talk about, first of all, is what is high resolution mass spectrometry, recognizing that not everyone in the room is a mass spectrometrist or even necessarily a chemist by training. Following up with what a typical experimental workflow might look like, with a bit of a caveat that it's half generalized and half about the way that MONet is standardizing their experimental approaches. And then at the end, I want to highlight a couple of case studies that showcase how real-world scientists have used this sort of data to make ecological interpretations. So what is mass spectrometry? It is the measurement of the mass of molecules. Not really, but that's a great approximation. In practice, we're measuring the mass to charge ratio of ions. And so what we're talking about here is that our molecules, our chemicals in our sample, they need to become ions. And so we have an ionization process. Most commonly, we're using something called electrospray ionization. It's described as soft, which means that most of our ions represent molecular ions. They're not fragmented. They are fairly representative of what the molecule looked like in the sample itself. But there's other ionization techniques, and every technique has pros and cons and biases. And there's also a big range of mass spectrometers. You've got low resolution instruments like single quadrupoles. You've got time of flight instruments you might have heard of before. But what we're really interested in today are FTMS instruments, specifically orbitrap or ion cyclotron resonance mass spectrometers. So with high resolution mass spectrometry, we're able to resolve species that have nominally the same mass. So two molecules that may weigh 370 Daltons, but they have a slight difference in their exact mass. And so these are described as isobaric molecules. And so, for example, there's two compounds here, a C16 molecule and a C13 molecule, that happen to weigh approximately the same amount. But with high resolution mass spectrometry, we're able to resolve this difference accurately and cleanly. And this difference is just on the order of about 20 millidaltons of salt. And so there's a couple of photos here showing you what a typical instruments might look like. On the left-hand side is an orbitrap coupled with a LC system. And on the right-hand side is EMSL's flagship mass spectrometer, our 21 Tesla FTICR mass spectrometer. So when we're talking about how high is high resolution, here's two spectra from two different high resolution instruments. On the top is a spectrum from our 7 Tesla CIMAX instrument. This is the one that we're using for the MONet project. And on the bottom is from an Exploris orbitrap mass spectrometer. This is the same sample, a swanner fulvic acid. It's a organic matter standard we use routinely. And you can see that across the whole range from about 200 to 600 mass to charge units, we see peaks across the whole breadth of this range. If we zoom in to just a single nominal mass, so this is 401 to 401.2, there's a dozen or so features, and each of those features is a distinct different ion or mass that we're measuring. And you can see just like really superficially and really simply that on the bottom with our orbitrap spectrum, we are getting a high resolution spectrum, but in this top panel, we're getting much higher resolution using our 7 Tesla ICR instrument. And what's really important to highlight is that every one of these peaks is a different species. It's a different molecule with a different molecular formula. And so we're able to routinely detect tens of thousands of distinct species in our organic matter samples across this wide range of masses. We can go even more extreme in terms of mass accuracy, in terms of how accurately we are measuring these masses and how fine splitting we can go with the distance between them. So from our 21 Tesla instrument, we can go to even higher mass to charge ratios and resolve even smaller differences. Here, the distance between these two peaks is on the order of 3 millidaltons, which is just about the mass of 6 electrons. So we're talking about really small differences in mass that we're able to resolve and assign different molecular formula to. You notice that we've got this MMA, which is mass measurement accuracy. Normally, if you're used to mass spectrometry, you might think in parts per million, which is a relative error. Here, we're able to achieve parts per billion mass accuracy. So we're talking about really good confidence in the measurement of the masses that we're getting. But we're not really interested in the real world, in the application side, in just the mass number. We want to know what that possible molecular formula is. And so how do you get a formula from an exact mass? The first thing to know is that every isotope has a distinct exact mass. So what this means is that there's a value after the decimal place, which isn't zero, with the exception of carbon-12, which we define to be 12.0. And so all of these other isotopes you see here have distinct, different exact masses. And what we're able to do is just essentially a combinatorial equation where we're looking at what possible combination of isotopes give us the mass that we've measured. Obviously, we constrain that with things like what's the natural abundance of those isotopes. You know, hydrogen-1 is 99.99% abundant. So we're probably going to factor that one in, but deuterium is only 0.11%. So we're probably not going to factor that in as much. What is really important to stress, though, is when we get a molecular formula assignment, we've got intact mass measurement. We don't have structure. So this page here represents 8 different structures that all have the same molecular formula. And so when we measure and assign a molecular formula to a peak, we're saying we're pretty confident it's that formula. It could be any of these structures, it could be all of these structures at the same time, or it could be structures that aren't represented here at all. And so it's really important to have this delineation in your mind of what we can tell and what we can't tell. So with that in mind, I want to talk about a typical experimental workflow, highlighting the steps from, getting your experiment designed all the way through to writing your paper and interpreting your data. So you've seen this already. I don't need to go into too many of the specific points. Suffice it to say that there's sort of a path that you follow when you do an experiment. The first thing is, of course, designing the experiment. Then you've got your sampling stages, sample preparation. Then we do our mass measurements. Then we have data processing, and then we have data analysis and interpretation. These last 2 sections are the focus of the tutorial this afternoon. But for the sake of completion, I'm going to go through each of these in turn. So when we're talking about experiment design, the first thing is obviously, well, what sites are we going to sample? We're going out into the field to get soil organic matter. There's soil outside the room right here, but presumably we're interested in something more specific. And so it's thinking about what soil types we're looking at. If we're going to predefined sites like neon sites, or if we're interested in specific geologies. On the right is a map of the sites MONet has already got proposals in for sites that we're sampling. And you can see there's a diverse range of the US that's being covered in different geologies and different environments. Of course, there's conditions we're interested in, time series, seasonal effects, whether people are interested in interventions like crop rotations on agricultural sites or irrigation effects, or irregular events. So what happens when there's a biomass burning event? What happens if there's a flood or a drought? And then you've got to think about your hypothesis. And I know this might seem a bit left field or a bit obvious, but it's so important all the way through the pipeline to contextualize your question to make sure that you're doing the right measurements and the right samples. And then finally, a little note is we often talk about replicates and sample numbers, and obviously more is better, but infinite is impossible. So you've got to figure out what your compromise is in terms of how many biological or field samples do you need, and how many measurement or technical replicates do you need. Recognizing, for those who aren't familiar with mass spectrometry, that there are challenges with reproducibility or batch effects. And so we need to make sure that we have the right number of controls, the right number of measurements when we want to aggregate data across experiments. So sampling, I don't think I need to tell anyone in the room how you go and sample a soil site, but you can find out more. MONet has developed these sampling kits. You'll go to the field, you'll get your core, and you'll put it in a little cylinder and send it to us. I won't go beyond that. Sample preparation, all of MONet's protocols are available online. This link isn't super helpful, but you can take a screenshot or look at this later. But what we do essentially is we take our cores, split them into sections, either top or bottom or top, middle and bottom. then sieve and air dry them. Then we do an extraction with water. Now, this is one of the most critical steps. When we're doing our mass spectrometry measurement, we're measuring a liquid. We're measuring the extract, whether that's the water extract or a different solvent extract. Anyone with any chemistry knows that like dissolves like. And so if we use water to extract organic matter, we're only going to extract a certain fraction of the organic matter, which is soluble in water and isn't so tightly bound to minerals that it doesn't go into solution. Different solvent extractions will give you different results. And so it's important to keep that in mind, again, in the context of your original experimental question. Likewise, we have to often clean up our samples using a technique called solid phase extraction. Wow, I'm not speeding up as I thought I would do. And so what's important to know with SPE is that it is also biasing in our samples. So the SPE is essentially like a little bit of a chromatography column itself, and some analytes will be retained well and some won't. And one of the most important things to note is that we typically lose very polar small molecules. So if you're really interested in primary metabolites and organic acids, those don't tend to be retained well by the protocols that we use. And so you've got to, again, keep that in mind when you're thinking about the types of carbon you want to study. And then we do our measurements. So we put our little sample in a little vial. We have infusion carts that automatically sample it. So we can run on the order of 50 to 100 samples a day. And then this is just another picture of our instrument. One thing to note, our typical measurement time is about 25 minutes or so per measurement. And we typically use about 50 microliters or so at a concentration of 20 ppm, which works out as only using about 1 microgram of carbon per measurement. But obviously, we can't just start with 1 microgram because of losses throughout everything that we do. Then this will go relatively straightforward, but we get our data from the instrument, raw data. It goes through CoreMS, which is a software we've developed here in EMSL. This allows us to do basically fully automated data processing, including signal processing steps, peak picking, formula assignment, mass recalibration as required. And then the output of that will be our table of peaks and masses and so on per sample. Again, this will be discussed a little bit more this afternoon. And important to note, MONet will be doing this step for you. But of course, the raw data will be available should you want to do this yourself. And then probably the most interesting thing for the people in the room, which is why I've left so much time for it, is the data analysis and interpretation. So we've got our mass spectrum, we measure a mass and we can assign a molecular formula to it. But then what we can do beyond that is what we call stoichiometric analysis. So we're looking at the ratio of elements in that formula and what we can infer about the chemical properties based upon that. So at the most superficial level, we've just got the ratio of hydrogens to carbons or oxygens to carbons. That can tell us a little bit about the degree of saturation or the degree of oxidation of a molecule. But there's other properties like double bond equivalence or nominal oxidation state of carbon, which we can use. Again, these will get discussed in a lot more detail this afternoon. And then we can do some basic visualization. So on the top right-hand side is a count plot showing the distribution of oxygen numbers per elemental formula that we're assigning. So here you can see that most of the species that we're assigning in this particular case have between about 8 and 15 oxygens in each molecule. And then on the bottom is a Van Crevelen diagram, which I don't want to get into too much just now, but suffice it to say, each formula we assign in a spectrum, we can then represent in this two-dimensional scatter plot according to its oxygen to carbon ratio and hydrogen to carbon ratio. And based on its position in this scatter plot, we can make inferences about the type of molecule that is, the type of chemistry that it might have. You can go beyond that, of course. You can do really sophisticated statistical analysis and interpretation, especially when you've got larger sample sets. This is really where the strength of MONet is going to shine as the data sets grow and evolve. So this is about a 10-year-old case study, and it's from Sweden, but they did this really cool experiment where they went out and sampled 120 different lakes, and they captured all the organic matter in these systems. And they did a combination of ordination analysis and correlation analysis relating key physical properties like pH or conductivity with things that they had measured with their high-resolution mass spectrometry. And they're able to make all sorts of really neat inferences about what that might look like. If you want more information, there's a review that came out about 2 years ago now, which highlighted basically what I've talked about. And so I strongly suggest that you have a look at that. It's really good sort of introduction to the state of the art and where future work needs to go. Similarly, self-citation. There was a book chapter that came out a few years ago, which covers the same sort of thing, but in the context of dissolved organic matter characterization. So there's nuances and differences between dissolved organic matter and soil organic matter workflows, but there's also a lot of overlap in the end. So we've gone through our typical experimental workflow, which again, and I know I sound boring, but it's really important to keep the experiment design and your original question in mind all the way through, because as I've tried to allude to, every single step you do is biasing the data in some regard. One of the really important things is that MONet is doing standardized workflows. So we're standardizing our sample extraction and our sample preparation. We're standardizing our measurements, but there's obviously the beginning and end of this workflow, which is kind of the you side of it, where you're deciding what sites you want to sample, you're deciding how to interpret the data you get out at the end. So briefly, there's 2 case studies that I think are worth highlighting. There's obviously way more than that, but there's 2 I have time for. The first one is the 1,000 Soils project, which was sort of the prequel to MONet. Now, this paper out in Frontiers in Soil Science really sort of served as an example of the things you can do with data, but as a really like powerful demonstration of real samples. So as an example, they had 3 cores from the WSU site, which had experienced different degrees of burning. And they managed to use FGICR and the characterization of the species present to make inferences about the different types of carbon, and specifically the different thermodynamic properties of carbon if the site had experienced a burn event, or if it experienced a lower severity burn, or if it experienced no burn at all. And then another example, which was looking at floodplains. And again, this thermodynamic inferences that can be made. And so here you're thinking about large biomolecules like lignin, cellulose that are in a soil core. But if the molecules and the microbes and everything that's going on are in oxic environments or anoxic environments, how does that change the nature of the carbon after time? And so what they observed here was that when you had oxic conditions, you had more reduced carbon, which I always get my head spun around on this, but essentially they could use NOSC, which is one of these properties we've already mentioned, and they could see statistically significant differences in the nature of carbon between these 2 environments. And so with that, I think we're back on schedule, unless there's lots and lots of questions.
Thank you, Will, for an amazing presentation and for putting us back to schedule. Will be here back also for the tutorial. So if there is any quick question, we'll take it now. If not, just collect your million questions in a Word document and we'll ask Will when he does the tutorial part. Yep.
I have a quick question. So why do you choose 7 Tesla and not 9 or 12? Because many papers these days, they use 9 or 12.
Yes. So one of the things I debated was how much mass spec technology to discuss. There's at one level, bigger magnets are better. There's an intrinsic physics associated with that. But there's also everything else that goes into it, including like the instrument design, the electronics that are associated with all of these things also have an influence on our sensitivity and on our resolution. And so what actually happens is that our 7 Tesla is able to achieve resolving powers like a 14 Tesla because of some clever tricks that go on in the electronics. It's also thinking about how do we design an experiment that can go for years and use the same instrument as far as possible. And so we have our 21 Tesla instrument, but that's essentially a unique instrument. There's no, like, if something breaks, we're not able to just buy a new replacement and swap it out. Whereas for the 7 Tesla instrument, it's as close to a modern commercial instrument as you can get, which means that when we're thinking about how do we standardize this and how do we get the best data, but also the most secure future, that ends up being our strongest platform to use.